Okay, so we're going to step through a number of aspects of the field of neurotechnology and specifically related to EEG. A little bit about our company, Advanced Brain Monitoring. We are a medical device manufacturer. We've been in business now for over 20 years, and over that period, we introduced 7 FDA cleared and CE medical marked systems. And we pioneered the development of systems that were mobile, scalable, easy to use. And so frequently they're used in home. And those technologies allow us to non-invasively measure brain's electrical activity, muscle activity, heart activity, respiration. I like to say that just about anything that you can measure non-invasively, we have developed some both sensors and algorithms for addressing that. We have a pretty large patent portfolio and a large customer base, and we've been able to diversify revenues through selling devices, software. More recently, we've done partnerships with pharma and took a leading role in providing clinical trial services. And we have received a little over 37 million in funds primarily from our National Institutes of Health, but also from DARPA and several other agencies. And this has allowed us to explore many novel applications for these technologies, both medical and non-medical. So I'm going to go through quickly and show you some of those applications. Next slide. First, well, let's take a step back in history. EEG, as you probably know, is over a century old. So it's one of our oldest known recorded physiological signals. And this, we borrowed some of Google's Ngram to trace the interest in EEG. And you can see, starting in 1940 and then peaking around late 1970s. You see many mentions in the Google text corpora, and then we see a decline. Now, what's interesting is if you look at neurotechnology, that those mentions have increased and coordinate or coincide pretty nicely with the second peak in interest in EEG, which has been driven largely by many of the developments in the field of neurotechnology. On the right, This is my undergraduate work at Ohio State. And you can see I'm surrounded by a room full of EEG equipment. And this is what we used in the late 70s. We've come a long way since then. But I just wanted to show this picture because it coincides nicely with this peak in interest. And I can tell you, we were so excited about the possibilities in the late 70s of what we could do with EEG. But it took the field of neurotechnology advancing, the advances in microelectronics, in battery, in computer processing, in wireless technology. We needed all of that as the platform to really launch us into novel applications. Next slide. And this is not just about our technology, although obviously I have a soft spot in my heart for it. There's been many new technologies introduced using EEG. These are 3, I think, that have received quite a bit of attention for good reason. The Zito really drew a lot of attention last year as being the first FDA-cleared dry sensor system, meaning that you don't require any kind of gel or scalp prep. You basically just put the system on and you're good to go. It's still a little cumbersome, but it is the first. BrainScope, I think, has developed and FDA cleared a whole set of novel technologies that are very impressive. Everything is mobile and fieldable. This was designed first with funding from DoD to be used on the battlefield for traumatic brain injury assessment, so that it's a novel sensor. You put it on the field, you don't have to know anything about EEG, and you have a handheld device that gives you the triages, helps you triage for traumatic brain injury. And the Cerebell is even a more recent FDA cleared device. Again, a very simple headband that has 8 EEG sensors. Anybody can apply it. What's really novel about their analysis tool is that it translates the EEG waveform into an acoustic wave, and you can then listen to it. And what they found is that if you're having a seizure or if there's a very abnormal EEG pattern, the acoustic wave sounds very, very different. So now you can have someone in an emergency room vehicle or anywhere put the system on really quickly and listen to hear whether there are abnormal EEG sounds. So these are just 3 of the novel medical systems, all FDA cleared, and that have very much expanded some of the applications for EEG. Next slide. And then there have been a number of other advances in dry systems, mobile systems, wireless systems. Wearable sensing was one of the first dry systems to really be adapted for widespread use. Cognionics right behind them. Cognionics has a fairly dense array system, again, all dry. Neuroelectrics I like to mention because they, to my knowledge, were the first to integrate the combination of EEG recording with the option of stimulating or neuromodulation through TDCS, TACS. So they've actually integrated into one design, one CAP design and one integrated system, both stimulation and assessment. And again, I think this is a very novel, combined therapeutic diagnostic system. And GTech, I mentioned because they are wireless. They were one of the first wireless mobile systems and have been used for many brain-computer interface applications. And then on the next slide, I'm only showing 3, but there are dozens, and I can barely keep pace anymore with the number of consumer devices that have been introduced that incorporate some measuring of EEG. NeuroSky was one of the older ones. More recently, we've had Muse. Emotive has been around for some period of time. And all of these systems feature obviously gorgeous designs, relatively simple recording, and they're really primarily geared towards either entertainment or relaxation, meditation, fairly simplistic applications, but being adopted and more widespread. And then this is our system, wireless, very lightweight. The entire system only weighs 3 ounces. So we've now, everything that you saw in my picture that was in that room is now incorporated into this small box on the back of the head. It's comfortable for hours. People forget they're wearing it. So again, incorporating all of these features of neurotechnology, we've been able to really change the way EEG is done. Next slide. There's also been many, many advances, and I don't have time to go through all of them, but software platforms, analytical techniques, methods for integration across sensing systems and across people, co-located or remote. And many of these platforms are freely available. EEG Lab, of course, is the workhorse coming out of UCSD, and they continue to upgrade it. And many, many people have been able to do EEG analysis using some of these platform systems. So this is just a brief list of some of the applications for EEG that we've been involved with. Biomarkers for neurodegenerative and psychiatric disease. This is an area that we are very excited about, and my colleague Amir is going to really drill down on what we're doing and what some of these applications are for EEG-based biomarkers. The field of neuromarketing, you're probably familiar with, has grown dramatically. We've done a lot of work in what we call team neurodynamics, where we're now looking. We've got 6 or 10 people with headsets on, and we're looking across brains to see how brains interact and coordinate during team activities. And of course, brain-computer interface has been a topic of the CNT from the beginning. And then human performance enhancement, we've done quite a lot of work in that area, and fatigue management, as well as occupational safety. Next slide. And again, Amir's going to drill down on this a bit more, but EEG is really very suitable for early detection biomarkers for dementia. Again, it's inexpensive, it's easy to apply. There's high test-retest reliability, and you can easily train people with no training in neurophysiology to acquire high quality EEG in a single half day session. Our goal here is to provide objective and quantitative profiles of brain health. So we envision a day where you will receive a daytime EEG assessment, just like you might receive a colonoscopy. So particularly if you have a history either of mental illness or you have a history of neurodegenerative disease, you may be required to take a brain health test where, again, EEG is perfectly suited for that. Next slide. Then we also have our whole series of sleep biomarkers, again, all using very simple sensor systems that can be easily self-applied at home. And for a lot of the studies that we've done, we're combining the sleep architecture and the sleep EEG with the daytime EEG for assessment purposes. Next slide. And we've done work with interactions between robots, interactions with prosthetics, and interactions with other smart devices. I think that, you know, the sky's the limit here, in that we can do so much translation of the EEG in real time now, and interface with, you know, other smart devices. Next slide. We've also been involved with neurorehabilitation, closed loop systems, or providing feedback to the therapist for stroke, spinal cord injury, and traumatic brain injury. And on the next slide, some of our recent work has been integrating virtual reality with EEG. And there we can do medical interventions and assess how the VR intervention is affecting the brain. And then we're involved in some educational as well as entertainment applications of virtual reality. And next slide. And as I mentioned, the field of consumer neuroscience or neuromarketing, where we evaluate consumer responses to advertising, product placement. You may have participants in a shopping mall or in a grocery store, or to evaluate product usability. And we found that you can use EEG metrics to predict whether someone is going to make a purchase decision or not. So pretty powerful methods for neuromarketing. And then we've also done work with peak performance training for golf and basketball, archery and marksmanship. Again, using a really simple system with just 4 EEG sensors, giving feedback to help people reach the peak performance state. And the last slide for me is about the team neurodynamics, where again, we're looking at EEG-based measures of cognition. We've identified EEG measures that predict emergence of leadership. And then you want to use some feedback of those metrics to optimize performance. And our most recent work with DARPA is showing the effects of social media messaging on both individuals and teams, and how social media messaging changes people's decision making. And these are things that we can see now in near real time. So I'm going to turn over the presentation to Amir, and he's going to drill down a little bit more on the field of EEG as biomarkers for neurodegenerative and psychiatric disease. Thank you.
Thank you, Chris. Very exciting. Hello again, everyone. So yes, I will be talking a little bit about the EEG biomarkers that is one of the areas that we have been active in the past few years. So basically, in terms of what are the applications in which EEG could be a very good biomarkers, it's whenever you have a challenge in quantifying a disease progression or treatment effect, EEG could be a very good candidate for biomarker. And I will talk about two of such examples. One is the dementia research and one is psychiatry research. And these two for different reasons. In dementia research, there is usually a very long delay between the start of the pathological changes that happen in the brain and the presence of the clinical symptoms. And in psychiatry, there's usually not a good objective measure or biomarker for cognitive decline, for the disease, or for, let's say, for depressive disorders, things like that. And so they're mainly subjective. And also in some applications, the EEG is not necessarily an indication of a disease progression, but we can measure the effect of the pharmacological compounds on the central nervous system, basically an objective measure to indicate that the drug is working. Now, whether that's effective in terms of the treatment or not, that's another issue. But even that aspect is very valuable for pharmacological companies where they want to see the effect of the drug, the compound, on the central nervous system. And this is all basically feasible because despite between subject variabilities that exist in EEG, it has been shown that within subject, we have a very nice test and retest reliability. So it's a reliable measure that we can use for quantifying either the disease progression or the effect of medication on human brain. There are obviously many different methodologies for analysis of EEG signals, as many as the signal processing techniques that you can imagine. But basically, we are analyzing the EEG signals in three different ways. One is the simple background EEG, you can call it, which is just resting state, no mental activity with eyes closed or eyes open. And the other one is when you are looking for evoked patterns in EEG, and that's when your participants or your subjects or patients are engaged in an activity, usually a cognitive test or something like that, in which you are having repeated trials and you're looking for evoked patterns, such as the ventilated potentials, the steady state evoked potentials, and things like that. And finally, you can use source localization using the EEG measures on the scalp. You can basically solve the inverse problem and identify what area of the cortex have been activated to give you that EEG pattern. So one example that I will be talking about today is Alzheimer's disease, and that's an area that we have been doing some research in the past few years. So in Alzheimer's disease, in most cases, there are decades sometimes between the start of the pathological changes in the brain and the start of the symptoms, which is in the form of cognitive decline and dementia. That usually starts with mild cognitive impairment and gradually goes into full dementia. And the point here is that the cognitive decline can be measured with neuropsychological tests. So these tests usually are not very sensitive, and by the time that you can measure this effect of the dementia, it's too late in terms of intervention. On the other hand, there are biomarkers that actually measure the molecular pathology of the Alzheimer's disease, the source of the problem, but they are usually very expensive and invasive. For example, a lumbar puncture to get actually the CSF biomarkers, and also usually it's too late. For example, there have been clinical trials recently that have been very successful in terms of eliminating the source of these proteins, such as beta amyloid from the brain, but you can still see that the cognitive decline is not eliminated. Now, what we are proposing here is to use EEG biomarkers, because as we know, these molecular pathologies, the way that they lead to the cognitive decline or to the symptom is through affecting the neural activity. So these neural dysfunctions are something that you can easily measure with EEG. So these EEG measures would be sensitive and inexpensive measure that are more directly linked with the symptoms rather than the pathology, although there are links between the pathology and the EEG as well, but it is less known at the current state of science that we are still looking at it. So this is an example of a very simple biomarkers by just looking at power spectral densities. In the y-axis, you see the power of the signal, and in the x-axis, you see the frequency. And then you can see that at the group level, we have 3 groups of Alzheimer's disease, mild cognitive impairment patients, and the healthy controls. And you can see there are very clear differences in the power spectral densities in these populations. And we have been working on quantifying these changes. For example, some measures that quantify the relation between the power in the lower frequencies, delta and theta band, and the higher frequencies such as alpha band, the ratio of these powers. And then you can see there are very significant differences at the group level between, let's say, healthy controls here on the left and the Alzheimer's patients on the right, and so also for MCI. And it's amazing how just 5 minutes of resting state EEG data background activity can distinguish nicely between the Alzheimer's brain and the healthy brain. And moreover, these measures can be helpful in terms of localizations of these abnormal activities, even though EEG has a relatively poor spatial resolution. But just looking at the channels of EEG data that are affected, we were able to identify that at least in our patient population, in the mild cognitive impairment, you can see that these increase in power spectrum in the theta band and decrease in the coherence and the connectivity between the channels in the alpha band start at the temporal regions. And then when we go to the full Alzheimer's dementia, these changes, these increase in theta power and decrease in alpha coherence are so widespread across the brain that it's very obvious and very significant. So especially the right temporal area in our population was a very significant area which had the most effect visible. And this is another example of our work that we try to model the cognitive decline in a longitudinal study where we basically model the change in what's called MMSE score, a measure of a very insensitive but still specific measure of cognitive decline, one of the neuropsychological tests. And we were able to predict this change in MMSE after 1 year of follow-up study, just using the age and the changes in EEG and the initial value of their MMSE score. Here you can see in the y-axis, you see the predicted value of the decline using EEG, and in the x-axis, you see the actual decline that happened in that score of cognitive decline. And it's very well correlated even in healthy controls who had a little bit of change in cognition. You can see that generally there's a good correlation here. And then you can see this score presented this in the AAIC conference this year. And finally, we can use these EEG measures for some sort of differential diagnosis, and we are in the progress of demonstrating that these EEG measures, to some extent, are specific to these conditions. For example, the EEG measures for Alzheimer's disease versus the Lewy body or Parkinson's disease, they have different patterns of activities. Here you can just simply see the theta band, alpha band, and beta band. These changes are different. For example, increase in beta activity is one of the hallmarks of the Parkinson's disease, whereas the increase in theta power is related to Alzheimer's disease. So this was basically a very brief summary of the research in the domain of Alzheimer's and dementia. But another example, which is also very interesting and very exciting, is the use of EEG as a biomarker of depressive disorder. For example, it has been very well established that the depressed patients or depressed individuals have preferential biases for recognition or detection of faces that are emotional, so sad faces with respect to the neutral faces. But these biases are subjective, and we have been able to use these biases using EEG, and in this case, this is not resting state, but event-related potentials. There is a component in event-related potentials around 170 milliseconds after you see a face. Obviously, it's not voluntary, right? So it's involuntary. And this component is being shown to be sensitive to detection of faces. And when we did this in a study, we were able to identify these and measure these components, this early N170 component. And as you can see here, in the depressed population, there is a very clear, significant difference between the sad and neutral faces when the participants look at these images to search for a target image, whereas in the control group, there is no biases that you can measure. So this work, we actually demonstrated in collaboration with Johnson & Johnson that a novel treatment for major depressive disorder actually affected these biomarkers, and in that way we were able to demonstrate the treatment effect in that trial. So generally, now the question is, what does it take for translation of these methods into the clinical practice? Because in many cases, the focus, obviously, for good reason, in the research community is focused on group differences and to demonstrate that there is an effect. But in my opinion, one of the next steps to actually translate into clinical practice is to demonstrate the effectiveness of these measures at an individual level, not just at the group level. And we have been working on this very actively. But even if you talk about the group level differences, still these group level differences would be very helpful in clinical trials where you have a control group and a treatment group and you want to demonstrate the treatment effect. And that has been actually increasing very much in past decades. In this image, you can see worldwide before 2000, before year 2000, according to the US National Library of Medicine and clinicaltrials.gov, you can see that there were only 28 studies that had the term EEG in their description with any indication. And this number from year 2000 to 2010 increased to 441. And now guess what? Between 2010 and 2020, this number is now jumped to 2,300 trials. Granted that this has not been normalized with respect to the actual number of trials, but we are seeing a very, very nice and good increase in using EEG for clinical trials, which we have actually been involved in ABM at several different trials that so with that, I would like to thank you very much for attending this meeting. And if there's any question for me or Chris, we would be happy to answer.
Any questions?
I have a general question. Can you hear me?
Sure, sure.
Yeah. So Chris and Amir, you did a great job presenting the overview of history of EEG hardware, software, and also applications. In these three areas, hardware, software, and application. Sorry. Okay. In these three areas, what do you see in the next 10 years? What would happen, or what would you like to see is going to happen in the next 10 years?
Please.
So I think all three areas are equally important.
Yeah.
So one thing that's already revolutionized the hardware is the Texas Instruments introduced their EEG on a chip. So now we have a chip that has 8 channels of EEG, and that tremendously reduced the size and allowed us to, our previous systems, we had to build the analog side, the A to D, each channel had an A to D converter, all of that is on the chip. Okay, I hope soon we will see something in batteries that is equally revolutionary. We have the lithium ion, it is rechargeable, but it's still, the battery is usually the largest component on our boards still.
Okay.
So I know Elon and a number of other people, the Al Mann Foundation. So batteries are important. For the implantable devices, they use charging, remote radio frequency charging. So we might be able to do something like that. I've always envisioned that the next EEG sensor would be, basically a Band-Aid, right? You take the adhesive off, you stick it on. It's got the chip in there. It's got, hopefully a rechargeable battery that is done via Wi-Fi. So you don't even really notice that you're wearing it. I don't think we're that far away from that. The processes, the algorithms, we've made tremendous strides, and we can do a lot more in real time or near real time than we could 10 years ago. And I expect we're going to completely revolution. I mean, maybe Amir would have some more comments on that, but I mean, even now when if we're trying to look at 4 or 5 people simultaneously and we're looking at coherence across brains, it's really challenging to do that analysis in real time, to allow you to give feedback to a team. But I don't think we're that far away from that kind of processing capability. And then, I mean, the great thing about the consumer EEG products is that they've kind of opened up this sort of what I call the democracy of EEG, the democratization of EEG, so that many people now have actually been able to see their brain activity. They're still trying to figure out a little bit, what do we do with it? But I think that is really changed the way society views EEG. We have finally started to change the way neurologists and psychiatrists see EEG, which the old school was, it's either sleep or epilepsy, that's all we do with EEG, period. And I think that, some of the work that Amir's doing and that he just showed is changing, the opinions or the use cases. So, so all of those things.
Yeah.
But Amir, do you want to talk more about analysis and software and?
Yeah, I mean, all of the points that you mentioned are great point. In terms of the analysis on software, it's many of these analysis techniques are not limited to EEG, right? So basically, the advances in signal processing is happening as we speak, and every now and then there's a new hype. Like right now, for example, there's a deep learning and things like that. But I don't think that's the actual hole in the picture, or the actual need for us. One thing on top of everything that Chris mentioned, for what I'm doing, I would like to see more is more interest in these analytical approach as opposed to traditional EEG reading in clinics, for clinicians. So neurologists, even as today, many are having that traditional mindset of EEG as being something to be just taken, not very specific, and just reading the EEG visually, and they can't really validate the quantitative measures, and some of them basically for good reasons. But we have been able to actually grow more interest in by we, I mean the community. For example, I've been attending AAN conferences and the Alzheimer's conference, AAIC conference, in the past few years, and there is a very big jump. For example, in AAIC, the number of papers that present or posters presented in EEG in this year was astonishingly more than last year's. Usually it was just a few paper here and there, but that's something that I would like to see for doctors and clinicians to at least have an open mind, trying to see for themselves what we can show with quantitative analysis of EEG for this clinical applications.
Yeah, it is, sometimes it's hard to change their mind, but I think it's coming. Yeah.
Yes, but again, as I said, the easy low-hanging fruit basically is clinical trials in which all you need to do is to show that there are some significant differences between treatment group and controls, and that has been very well accepted by pharma companies, and yeah, that's good.
Thank you.
Thank you.
I have a quick question. I do want to jump on the topic where we were exploring, what additional applications can EEG provide for both the clinical and commercial space. And my question is, do you think that there's going to be some sort of upper limits to what EEG can eventually be used for in terms of its applications because of the limited spectral bandwidth that we can really look at, or like potential temporal or spatial frequency? I kind of wanted your thoughts on that.
Sure. I don't want to suggest that EEG is a final diagnostic for all, brain disorders, or is useful in all situations. There's always going to be limitations. First of all, it's a very tiny signal, and there is a lot of noise that you have to identify and decontaminate. Biological noise, there's electronic noise, and I think we've had 100 years to work on these problems, so we've gotten pretty good at that. But then it is always going to be a reflection of the summated potentials of neuronal connections. It's not going to tell you exactly where something is going on in the brain. As Amir stated, in our mild cognitive impairment, we do see, that the EEG changes first over the temporal parietal regions, which makes sense because we're talking about, hippocampal circuits, and you're going to tap into reflections of those, and those correlate to the EEG we've shown correlates with FDG PET. It correlates with hippocampal atrophy, some of the more the other measures that we have access to. So it's only going to give you, a certain level of data and level of analysis. But I think that we've shown, through different analytical approaches, that there's a lot more data to be mined in the EEG than we've accomplished to date. And, it's unfortunate, with EGI doing the very dense arrays, that Philips wasn't able to, create some of the products that I think they had originally envisioned, because there you have access to, many, many, many more sensor sites. And, that gives us additional information in terms of source localization. But it's always going to be an estimate. And I think, but the beauty of EEG is it's easy and it's inexpensive, and it's going to get easier and less expensive. But certainly there's limitations, yes. Amir, do you have thoughts on that?
Yeah, I think you covered it all. And also I would like to point the keynote talk this morning was very interesting. And at the end, there were a couple of slides about what you can and can't expect from EEG or neurotechnology in general. In the near future, that would be a good point also for you to go back and.
Yeah, so if you want to localize a particular region of the brain, like an epileptic foci, EEG is helpful. If you want to find a tumor or a hematoma or some traumatic brain injury, obviously EEG is one tool among many others. So there's always going to be a combination of tools. But something like, we were fascinated with the emotional faces test, where for depression and for some of the other psychiatric diseases, there are very few objective quantitative measures. And here's something, I mean, it makes really good common sense that if you're depressed, you would overreact to a sad face in comparison to a neutral face. But we can actually measure that and quantify it. And with J&J, we were able to show that a novel compound for treating depression had an effect on mitigating that enhanced response to sad faces. So I think there's a lot of potential in psychiatry where you don't have any access to other objective tools for studying the brain.
Yeah, I definitely agree with that point. And I wanted to follow up by asking what kind of expected variation, expectation of variability between subjects or patients with these different either psychiatric disorders, or you guys mentioned Alzheimer's and dementia. I know that biomarkers for that seem to be fairly straightforward. And like I saw some of the figures, they were fairly like condensed. But with things like psychiatric disorders, there are lots of variability there. So what are your thoughts on that?
Do you want to comment on that, Amir?
Yeah, so of course, so that's something that to some extent is unavoidable. But for example, things like age, especially in our dementia research, age is a very important factor. So you always need to age match your study groups because they clearly affect your measures. And even things like in my past research, I've worked on steady state visual evoked potentials. You can see that even the folding of the cortex, different being different participants, could affect these signals. So there's always certain levels of subject variability, but there's always ways to tackle that, right? So usually we use relative measures of power spectral densities, so that even if you have differences in baseline, relatively speaking, how would these measures work? You can't eliminate that, obviously, but in my opinion, the best or the more elegant way to approach this problem is to include more dimensionality, to include other dimensions other than EEG. So if you have a model that it takes into account both the EEG and other clinical indications, other clinical measurements, genetic data, especially also in Alzheimer's, genetic data is very important. So if you think about a personalized medicine approach, if you have a large body of data sets that you can include all these different dimensions of the data and then start modeling, then one day you might be very successful in modeling these disease states or treatment effects or whatever based on everything. But currently, if you increase the dimensionality, then you don't have enough data points, right? So right. The way to do it is to have basically larger scale data sets where you can model these changes globally.
And again, the within subject reliability of EEG is quite high. So, and we've looked at some of our employees over many years, and the consistency in the power spectra is remarkable. That you can exactly the same alpha peak, exactly the same alternative peaks. If you have a beta peak or and even over long periods of time, that is very consistent within an individual. So it's, again, useful for a personalized medicine approach where you may want to try multiple interventions and see within an individual which one of these or which combination of these are the most effective. And the effect sizes between groups are fairly large, Amir, when, even the difference between mild cognitive impairment, which is kind of a catch-all category, and healthy controls is pretty substantial in some of the EEG metrics. So one of the things we've done is, you can use a lot of EEG features and then build some machine learning models to better discriminate, across individuals.
Thank you. Did anyone else have any other questions? Because I do have a couple more, but like I don't want.
Sure. I think there's only 3 persons left. So yeah, feel free to ask any question.
Just,
Okay.
sorry, just before you start, I have another meeting that I'm leaving now. So
Okay, sure.
yeah, sure.
It
Good
was
to see
nice
you,
seeing you,
Amir and
Rosa.
Chris.
Likewise, thank you. Talk to you later.
Bye.
Yeah.
Bye.
Okay. I was interested in your work with the VR space. Could you talk a little bit more about that? And what is it, more of a commercial facing development, or?
Wait, can you repeat the question?
Yeah, you guys mentioned that you are working with VR and integrating
yeah,
EEG with VR.
Yeah, so we've worked with a couple different VR headsets. We never really accomplished a completely integrated system, but I don't think we're far from doing that. The beauty is just about any of the VR goggles fit over our headset really well. And what we did was we built a whole neurocognitive test protocol inside VR. And because one of the things we wanted to do was just first look at some of the brain activity, both the resting state and some of these event-related potentials during attention and memory tasks, to see if there was any difference in the VR environment. And there really wasn't. So, it was interesting, maybe not what we were expecting. And then we started to look at, okay, so what if we do some interesting, stimulating gaming environments with people who have mild cognitive impairment? Will that, in VR, will that enhance the brain activity? And can we see it, in real time? And we haven't really done enough work yet to say it will, but I can tell you that almost everyone, no matter what age you are or what ability you are, enjoys doing things in VR. So that's kind of the first step. And then we've worked with a couple other, so we did work on one of the Facebook projects with the Oculus Rift and looked at people meeting each other in virtual reality for the first time and how they interact, and identified some neurometrics associated with introverts and extroverts, and how meeting in VR impacts that. So that to me is kind of the more fun aspect. I think it's just the beginning. One of the problems that we ran into was that getting people who do VR programming is not inexpensive. I mean, getting VR content, let's put it that way, getting your own VR content is not inexpensive. And you can time sync the EEG very easily with the VR. That's something that we learned quickly, but you need to be able to have full control over the content. And if you don't create the content, or if you don't partner with someone creating content, you don't have that same kind of control. So look, I think there's all sorts of exciting things that we can do in VR. We're talking with a group now about doing training for police officers, where we present them with evocative situations in VR and look at the brain responses and train people to, improve the way they evaluate a situation before they shoot. I think that's the kind of thing where the combination of the tools is really potentially exciting. Did you have a specific project you were interested in with VR?
So our lab recently moved towards the VR space, where we use invasive ECOG and work with epilepsy patients. Mm-hmm. So we have a really cool opportunity to actually just have this really high density recording, but eventually we want to transition that into EEG space as well. But I'm most interested in artificial sensory stimulation to provide feedback, sensory perception feedback. Mm-hmm. So an example would be if someone in VR space touches an object, they would be able to artificially feel that object as well. And I thought it was interesting that you guys mentioned, I think there was a company, Neuroelectrics, where they had an integrated EEG and stimulation system. And I was curious, like, what you guys think about having the recording, the stimulation, and the VR all like integrated into one headset. Like, is that even possible?
I don't see why not. I see no reason that you couldn't do all three things. Definitely not. I mean, you could have the VR going. The stimulation itself sometimes will disrupt the EEG for a period of time. So you won't necessarily get everything simultaneous, but there's no reason you couldn't try.
And just like a follow-up on that, I thought it was really cool how you mentioned that the EEG device, the patients or people who purchase this device can just put them on themselves. It requires very little calibration, which is the opposite of stimulation, where that requires immense calibration. So I think like trying to solve that problem is going to be one of the next steps towards like.
Yeah, and that's something that I think most of the wireless, easy to use systems have achieved in the last decade. You don't have to abrade the scalp. You don't have to, you know, there's a lot of things. You don't have to calibrate the system every time. And it's eliminated a lot of the things that we had to go through in the 70s to get high quality EEG. All right.
Thank you so much for coming here and presenting. Yeah, really appreciate it.
Great opportunity.
Thank you.
Nice meeting you.
It was nice to meet you.
Luck with
Thank you.
VR, stimulation, EEG. Yeah.
Thank you.
All right. Thank you.
Bye.
Bye.